Hey everyone, today we're talking about the materials and methods section. It can just be a methods section, meaning that you don't necessarily have to be specific about materials if you don't want to. You can just combine it all as one methods section. That's typically what I used to do when writing my papers. What you'll want to do, and this is different than what your poster would be, however, your poster can mirror this, is that you'll want to organize this section into subsections with headers for each procedure that you do. For example, if I were going to do a field collection of some uh, organism out in the wild and then do tests on them back in my lab, first I'd want to set up my procedure for the collecting, then the data part of it in the lab. I'll show you an example of this using a paper that Liz used for her inspiration for her project in just a moment here. But the aim of this section is not to step by step walk someone through the whole thing. It is to demonstrate that you knew what you were doing and that your reader could recreate your experiment, but it's only that to demonstrate that you are using scientifically valid methods, meaning that the things that you didn't have any holes in it, any problems with the procedure, didn't veer off in the direction that you didn't really intend, and to provide the reader with enough information to be able to do this themselves without being step one, gather materials, step two, blah, blah, blah. Okay. No more stepwise procedures. You guys are old enough to know that by now. And I've said it over and over for you guys, so you should be familiar with that. It is going to be in chronological order. So if you had to culture your yeast first and then do experiments on them, the first section would be yeast culture. The second section would be experimentation, stuff like that. You'll also want to include any statistical analyses or measurements that you performed during this time, as in if you, we're not going to tell them how to measure using a ruler or anything, but if you made some quantifiable variable, like Caitlin, for example, used a spectrometer to see how much area, or excuse me, how much transmittance was going through her yeast samples, she would have to additionally talk about the spectrometry there. If you were counting up the mass of something or looking at something under a microscope, again, those are things that you did to get the data that you're going to be referring on. So that is the thing that you'd want to actually cite. If you end up doing any statistics, you'll just mention it here. You won't actually show the statistics that you did, but you'll mention that you did a specific statistical analysis. Okay. If you followed a procedure from another paper or kind of piggybacked off something in the past, you should cite the source that it came from and provide a general description of the method overall. But you don't have to go in detail past what they did, as in try to infer what that previous paper did and set it up on your paper. Okay. You are essentially giving them the avenue of how you came to do the things that you did, but you don't necessarily need to repeat every word that they absolutely said. Finally, it'll be very hard for you to resist, and I want you to resist the urge to talk about any errors that happened, meaning that if you somehow didn't get any results after 24-hour and you decided to leave it in the incubator for 48 hours, or if you had one run where you accidentally had too old a hydrochloric acid, but you tried it again and it worked out fine. Don't talk about the things that went wrong. Things go wrong in the lab all the time. You don't necessarily have to talk about it in order to seem valid. Okay, uh, talking about your mistakes is great on a one-on-one conference with me or in uh, the realm of a reflection, like for your scholarship essay or video, or even when you're talking to people one on one at your presentation. Talking about your errors is a really great way to show that you learn from mistakes and you really learn from the project overall. But in the paper itself, you're not arguing that you did stuff right. You're arguing that you did something total. So if you talk about the stuff that you did wrong, you're kind of just filling up space to fill up space. And I'm sorry, you're going to have to resist that urge because You guys did a marvelous job on your projects, even if something went wrong. A lot of you even backtracked and said, okay, how can we go about this a different way? What is the error least common denominator that I need to solve? Okay. So make sure you don't talk about the things that you did wrong to lead you down a good path. Just talk about the good path. Okay. Now, I do want to show you that paper that I mentioned of how to organize this. So backing up here, going to this paper, and move this out of the way. There we go. This is one of the papers that Liz had me read to get the inspiration behind her particular projects. It was a paper that really looked at the differences in how cancer cells react to progesterone and three different other types of synthetic hormone therapies. Those are them. She's using two of these. Um, but if we go down to the methods section, in this abstract, they split it up into different sections, but I want to go down to the actual methods section here. You can see that they started the methods section with the cell culture and treatment, meaning that they had to specifically have the cell culture of the breast cancer cells that they used, and they talk about it very specifically about what they did. No, they didn't list it as a 1, 2, 3, 4 stepwise procedure, uh, but in this case, it is going to be different than the project, or the poster, because you are going to be in more detail this time around. So if you wrote something for your project proposal that you thought was detailed enough, you can probably do some copying and pasting here. You can also see that in this specific section, anytime that there's a material used or a chemical used or a piece of equipment. Like you can see, they got the progesterone, MDA, and 17 beta estriol here. They got it from Sigma Aldrich here in St. Louis. Now, I'm not going to make you put that in there. I think that's a little bit extra personally. But by all means, if you want to list the things that you used in the lab for the different parts of your paper, that's totally fine. So if you have specific things that we used for cell culture, like specific strains of yeast or specific uh, E. coli strains or something like that, you could list it in the section that you're talking about your procedure. Notice that after culture, this person went into details about their immunofluorescence, which is the act of washing them under a protein wash and getting them to glow in the dark so that you can see them better, and then blotting them on a machine. They went into further detail with each of their transfection experiments and migration assays to see exactly how these cells reacted to the treatments that they described all the way up here. In fact, they didn't, when Liz and I read this, they did not do a very good job of describing the actual treatment themselves. They did a better job looking at the figures that they had, the pictures that they have, which we couldn't even get to see because some reason it's not in this particular PDF version. But you can see they have everything separated very specifically by situation. In fact, if you choose to do this subcategorying in your method section, you can actually use the same subcategories or similar subcategories in your results section when you talk about the different pieces of data that you collected. Okay. All that to say, your materials sections, your method sections, are very straightforward and pretty easy to write. So hopefully you got a good idea of how you're going to organize it. Um, but it is really up to you at this point. The methods section is a way for you to show your chops, show what you did, and prove that you are the good scientist that I know you are. Totally up to you. Let me know if I can help you at all with that. That is the end of this introduction to the section here. There's not much more I can say about writing a method section. I'm glad I could give you an example here. But let me know if there's any way I can help. Good luck, be smart, and I'll see you tomorrow.